Hello, welcome everyone. Today I'm going to present one of my research projects that I've been doing over the summer in my job as a lab assistant in Dr. Kalischuk's lab, working in plant pathology in the plant agriculture department in the Bovie building. So this project is for screening Ontario strawberries for the presence of Neopestolopsis. And you may be wondering, what is Neopestolopsis? So it's actually a fungal pathogen that affects strawberry crops. And a new strain has actually been discovered in Florida in 2019, and it's caused over 80 hectares of strawberry crops just in Florida alone to be destroyed. So it was actually discovered once in Ontario last fall, which is not good. And our research goal is to determine if there are any other occurrences and if it's growing. So that's just what we're going to figure out. So looking at the material and methods section, specifically the first step. So the first step is initial screening of the strawberry leaves. So I'd get these strawberry leaves in, these samples, and I'd do just a preliminary check of them. I'd look under the microscope, see these different lesions, determine different morphology of this fungus, different traits, and see if I can determine if it's N-pest right there or not. And N-pest has this really cool structures called conidia, and they're actually located in a servula. So if a sample has N-pest, sometimes you can see these black, tiny little microscopic circles that just look like, they kind of look like dirt, but they're very circular. So you can look under them under the microscope and you'll see all these conidia. And this is just a photo I took, and that's just, you can determine by morphology of this conidia if, it's a good indication of N-pest. And when I talk about N-pest, I do mean Neopestolopsis. It's just short to say N-pest. So the second step is culturing. So I'll take these strawberry leaves. I'll perform a surface sterilization of these strawberry leaves because we don't want any fungus that is not already infected into the actual strawberry to show up in the cultures, because we just get a ton of funguses off the leaves. So just sterilize the leaves, cut a tiny little piece from the lesion, place it on this PDA agar plate, wait approximately 5 days, like 3 to 5 days for it to grow. And I typically do 3 to 4 plates per one sample, just in case that there's experimental error on my part. But also there's different types of funguses in different samples, so I just want to get a good broad look at the sample. So after these cultures grow, I put them and I perform a DNA extraction. So I go ahead and I take the DNA from these cultures, and then after that I run them through the PCR, through the thermocycler. So for the PCR, we're using specifically ITS and beta tubulin primers. So this amplifies this fungal DNA, so specific for fungal, like funguses, or fungi. And then we can determine from there. So after this PCR, I run them on the gel. I run these samples, the PCR products, on a gel. So what we're looking for is, we have the different ladders here, as you can see on the sides. Now we're looking for these bright marks here that shows that that the DNA has been amplified. So that means that there is presence of fungal DNA, which is good. We're looking for it. So after we do that, we're gonna, what kind of fungus is this? So we're gonna purify the DNA first, so we can send it off for sequencing at the genomics facility at the Summerlee Science Center. So they send me the sequences back, and then I put them through the BLAST, which is just an online software website that determines what kind of species it is. So looking at the results section, so these are my BLAST results of the strawberry fungi, the strawberry fungi. So we're just looking in this poster specifically as whether it was positive or negative for an N-pest result. And as you can see on the side here, we have 7 positive results out of 16, which is crazy, 7. So We did find NPest in Ontario, but also I looked at the phylogenetic tree of Neopestolopsis. So I actually created a phylogenetic tree using the sequences that I got from the genomics lab for the beta tubulin of all the different NPest positive samples. So I took those and I put them in using MEGA, which is a molecular evolutionary genetics analyst software. So I put them in there, align the sequences together, and then I made a phylogenetic tree out of that, which is pretty cool. So it just determines how close in similarity they are to each other in genetics. So it's pretty cool. So we're wondering, are they close to this really aggressive strain of NPest that was just found? And it was found, we did find that it was similar to this aggressive strain, which is not good at all. So yeah, that was the result of our of this research project. So yeah, I hope everyone liked my presentation. I just want to acknowledge my advisor, Melanie Kalischuk, and Katie for bringing me these samples, and I hope everyone has a great rest of your summer. Thanks.